Hello students, welcome to Aakash. I am your botany faculty, Dr. Sukirti. So today we are here to discuss the paper MT4 of AIATS Plus 2025 Phase 2, which was held on 9th of Feb 2025, right? So this particular paper is giving you a brief idea that how exactly you're going to see the NEET paper as well, right? A kind of a practice so that you can have a thorough understanding of all the topics as well. So in this session also, we are going to deal with the questions of botany so that you can understand that how to deal with the questions and at the same time know the entire periphery of the questions, that how exactly the questions could be asked in other ways as well and how to approach them, right? So not taking much of the time, let's start with the questions. The very first question over here is talking about giving certain statements and talking about that which ones are true for alleles, right? Now in order to understand the concept of allele, firstly we should know what is gene, right? Not the one that you're wearing, and of course the spelling is also different, but over here the gene that we are talking about is the stretch of DNA that's going to code for RNA, right? Further, it can code for protein too, right? So when we talk about this gene term, it's a broad term which is basically expressing a character, right? Now, the detailed term or different forms of a same gene would be considered as allele. Like for example, if we consider that there is a gene named as T, right? So if I'm writing capital T or small t, these will be the alleles, right? So as gene is expressing a character, which is again a broad term, a feature of an organism, similar way the alleles are going to code for traits, right? Traits are coming under the character, that means the different form of a same character. Like if I am considering plant height as a character, so tallness would be one trait and at the same time dwarfness will be another trait, right? So this is all about alleles, right? So the first statement says that it codes for a pair of contrasting traits, right? That's how the definition of allele emerges out, which says that it will be coding for a pair of contrasting traits. So if I am considering capital T allele, so it is going, and we are considering garden pea plant as an example. So this capital T is going to induce this particular phenotype, tallness in the pea plant. And on the contrary, if we talk about the small t allele, it is going to induce the dwarfness, right? So that is why we can clearly see that these are two contrasting characters. One is inducing a very tall height and another is inducing really small one, right? So yes, alleles do code for pair of contrasting trait, but yes, not always. Like if we see the case of, let's say, ABO blood group, right? So it has multiple alleles, right? It has multiple alleles. Over here, one is going to code for A blood group, another is going to induce B blood group. If they are together, they're going to induce AB blood group, and then in this particular thing in homozygous state is going to induce O blood group, right? So it may sometimes happen that they are not going to call for contrasting traits, but yes, they are going to show different forms of the same character, right? Different traits of the same character, right? But the statement over here is saying that code for a pair of contrasting traits, which is the usual case. They are not saying that always they're going to code for contrasting traits, right? So this statement will be considered as the correct one. Don't confuse it by taking the example of multiple alleles, right? Then it is saying that they are slightly different forms of the same gene, right? And because they are the slightly different form of the same gene, that is the reason why they are giving the different traits of the same character, right? So that is the reason why we are going to consider this statement as a true statement, because it is depicting that alleles are slightly different forms of the same gene, right? Now the third statement says true breeding line for a trait will have either both allele dominant or both allele recessive, right? So in order to understand that, firstly we need to know what is the true breeding line, right? Another name that we use for them is pure lines. And an important characteristic of these are that they show stable trait inheritance. That means in the entire line, that particular character is going to remain the same. Like for example, if we are considering a line of plants, right, of let's say Pisum sativum. So all the plants are basically tall, right? So then we are going to call that this particular line of plants is a true breeding line or a pure line. And in order to impart this particular, inheritance pattern, the plant or the organism should be homozygous in nature. Like for example, we'll understand it that why we are saying that it needs to be homozygous is because of the fact that, let's suppose again we are considering plant height. So if they are going to be homozygous dominant, right, then every time the progeny that they are going to have will have capital T, capital T. That means again the progeny that they are going to produce would be tall. Similar way, you can have any number of generation and every time the progeny will impart this particular genotype and induce the tallness in the plant, right? Similar way, if I am considering it to be homozygous recessive, then also what happens? That the type of progeny that we are going to see would be always small t, small t. That means the plant will remain dwarf in all the coming progenies too. So that is why pure lines show stable trait inheritance, and in order to start with it, right, we take the pure lines as the parents to start the cross, right? So now going back to the particular statement which says that true breeding line for a trait will have either both allele dominant or both allele recessive. So that is true. It is saying that the true breeding lines will be either homozygous dominant or homozygous recessive, right? So this is also true with respect to the allele. Then the fourth one is saying that both the alleles in a zygote are always contributed by one parent, right? So when we talk about the zygote, so we know that its formation is a resultant of fertilization, which is a characteristic event of sexual reproduction, right? Now in order to facilitate the process of fertilization, we need two different gametes from two different parents, right? And when they fuse, they are going to form zygote, right? And as both the gametes are basically fusing and contributing in the formation of zygote, that is the reason why that zygote will receive half of the allele from one parent and half of the allele from the other parent, right? So someone is asking, homozygous recessive is also pure line. So yes, homozygous recessive is also going to be pure line, because in the upcoming generations too, they are going to have this stable trait inheritance, that the progenies, if they keep crossing, like self-crossing, then they are going to generate the progenies that are going to be, showing the dwarf character or the recessive character. So that is why zygote is going to receive the alleles from both the parents, right? So as the particular statement is saying that both the alleles in a zygote are always contributed by one parent, so that is not true. And as the question is asking about the ones that are true for alleles, so those are A, B and C, correctly depicted in option 2. So that's how you're going to answer this particular question. Hope this is clear to everyone. So Xenia is saying that alleles or gene ke beech ka difference, okay. So I hope with this particular one you are clear with this, right? So now let's move ahead to the next question, and that is question number 132, which talks about the correct sequence of events in Prophase I. So this is a very important topic with respect to NEET examination, because n number of questions with different, twists are being asked from this particular topic, Prophase I. Certain times they'll give you the name of the subphases that occurs during this particular phase of meiosis I. Certain times they'll ask you about the sequences by giving the events name. So that's why it's important to understand that how exactly Prophase I is entirely exhibited, right? So let's understand what is Prophase I all about. So because when we talk about prophase, this is the first phase of the karyokinesis of meiosis I, so it will show certain characteristic events, like it will show condensation of chromosomal material, right? Then it will show nuclear membrane disintegration, and at the same time disappearance of nucleolus, right? So these are certain key events that we are going to see during prophase, right? And if we are going to talk about the animal cell, then aster formation is also going to take place. But then how it is different, right? How exactly this Prophase I is told to be the longest phase in meiosis and also different from Prophase II and the mitotic prophase, right? So for that, what we are going to see is the events, what are the unique events of Prophase I, right? So when we talk about the very first phase of Prophase I, that is leptotene, right? Leptotene being the starting phase. Am I audible to you? Is my voice audible to everyone? All right. So we'll start again with leptotene. So leptotene being the first phase, it will start showing the characteristic event, that is condensation of chromosomal material, right? So in this phase, it begins. So it will show the beginning of condensation of chromosomal material. One important thing to focus over here is that it's not going to end here. It will be continuing throughout the prophase, right? It just begins in this particular phase. Now when we move ahead to the next phase, that is zygotene, right? As the name itself suggests, zygotene, right? Similar to zygote sort of a thing. So over here what's going to happen is that the two homologous chromosomes that we see are going to pair together, right? Now in order to make them pair, what we need is a structure which is depicted over here in a ladder-like form that is known as synaptonemal complex. This ladder-like structure would facilitate the synapsis process, the key event of zygotene, right? So if someone asks you that where exactly the synapsis occurs, it will be zygotene. Now, after synapsis, the structure that you are going to observe would be a bivalent, right? Now, what is a bivalent? So, when we talk about two homologous chromosomes, right, and because now they are being paired together, with this ladder-like structure and now behaving as one unit, that's why we are going to call them as bivalent, because now we have two chromosomes together, and because the number of chromatids over here has now become 4, we can call this structure as tetrad 2. Right? So if the questions ask that what exactly where exactly the formation of tetrad take place, you have to mention zygote. But in this particular phase, they are not clearly visible as tetrads. In order to see them clearly as tetrads, we have to move towards to the next phase, that is Pecitine. So over here, the characteristic event of Pecitine phase is crossing over, right? Or we could say recombination. So in this particular phase, you can see these bivalents clearly as tetrad, but the main event is crossing over, recombination, which would occur between the non-sister chromatids of the homologous chromosomes, right? So if let's suppose we are considering them as the non-sister chromatids of homologous chromosome, right? So if we make a chromosome like this, right? So these two are considered as sister chromatids, right? The chromatids of the same chromosome. Now this one, let's suppose I'm naming them as A, B, C and D. So B and C, or A or C, the chromatids of a different one will be considered as non-sister chromatids. So the recombination is going to take place between the non-sister chromatids. But in order to facilitate this particular process, firstly we need an assembly that will allow a tight binding, right? So that particular assembly is known as recombination nodule, right? This is carrying the enzyme recombinase, which will allow the exchange of segments between the non-sister chromatids, right, of the homologous chromosomes. So because of this particular event, that is crossing over, what we see is variations in character, right? And that is why meiosis is being considered to bring out the variations in the organism, because of this particular characteristic event of crossing over, right? Now after this particular crossing over is being completed, then obviously the pairing part, the synaptonemal complex, is no longer required, right? Because in order to exchange the things, they have to be in close proximity. So when the crossing over is being complete, the cell will progress towards the next phase, that is the diplotin phase, where the characteristic event that's going to happen would be the dissolution of synaptonemal complex. Because of this particular one, what we are going to observe is that the chromosomes are getting separated from each other except at few points, right? Like for example, these chromosomes are now getting separated from each other. Let's suppose I'm making it as a different one so that we can understand that the crossing over has already been taken place. So this segment has been exchanged, but this segment is still attached with a particular structure, which is recombination nodule. So rest of the synaptonemal complex that's been initially present between these two chromosomes has now been dissolved in the diplotin phase, except at these points where crossing over has occurred. So these are the site of crossovers known as chiasmata. So this particular structure is going to be visible in the diplotin phase, not prior to it, right? After this diplotin is being finished, in certain oocytes of some vertebrates, what we are going to see that this particular phase retains for a very long period of time. But after the completion of this particular phase, now the cell will enter towards the dikinesis, the fifth stage, or we can say fifth substage of prophase I. So dikinesis is being characterized with the terminalization of chiasmata, right? Now, why this terminalization is being required is because if the chiasmata is not going to be removed out or terminalized, right, then these two homologous pair, chromosome pair, won't be able to separate from each other, and that's how we are not able to, give them equally to two daughter cells, right. So this can cause a chromosomal aberration in the resultant progeny. So that is the reason it's important that the terminalization of chiasmata occurs, so that this homologous pair could get separated from each other and could be equally distributed to the progeny cells. So this is what happens during diakinesis, but then till now the nuclear membrane disintegration is occurring. So that disintegration is going to be there, the nucleolus disappearance is going to be there, and the aster formation and all those events that are still lacking are going to be finished by the end of diakinesis, and therefore diakinesis represents the transition towards metaphase I, right, because this shows that the prophase is now completed and now the cell could progress towards metaphase I, right. So this is the entire idea about the prophase I. If you have any questions, you could put in the comment box so that I could answer. And prior to this, let's get back to the question. So over here the question is really simple, and it talks about that what is going to be the correct sequence of prophase I substages. So as we know that it is going to start with leptotene, right. So in all of the options it's leptotene, so we can't still rule out the options. Then leptotene, after condensation, there has to be a synapsis even that will be occurring, right, that occurs in the zygotene phase. And as option 2 and option 4 is depicting that after leptotene, pecitine or diplotene is going to be there. That's why we could rule out this particular options. Then zygotene is being followed by pecitine, where the crossing over takes place, right. So it's been here. Based on that, we could rule out option number 3, as it is saying that diplotene is going to follow zygotene. After pecitine and diplotene, we will be seeing that the crossing over has already been finished. Now this time we are going to have the formation of chiasmata and at the same time dissolution of synaptonemal complex. So formation occurs during zygotene and dissolution occurs during diplotene, and finally ending the prophase with diakinesis, right. So that's why option 1 is correctly depicting the entire events in sequence, right. Hope you understood it. All right. Now let's move to the next one. That's going to be question talking about the correct sequence with the stages of karyokinesis, right. So when we talk about karyokinesis, that means one of the phases of M phase, which begins with karyokinesis, that's the division of nucleus, and further cytokinesis, which is the division of cell. So it's just talks about the karyokinesis, right. So when we talk about karyokinesis, right, so it begins with the very first phase, which is prophase, right. So when we talk about prophase, as I have mentioned you, there will be certain events that's going to occur. Firstly, the condensation of the chromatin material, then it will be the nuclear membrane disintegration, and nucleolus also going to be disintegrated. Other cell organelles are also going to be disappearing from here. So these are certain characteristic event. If it's an animal cell, then we'll see the aster formation, which could be seen here. So what happens over here is that the centrosome is going to move towards different pole and then start radiating out microtubules, right. So do remember that certain time you got confused that chromatid moves to different poles and centrosome moves to different poles. Centrosome movement occurs during prophase, chromatid movements occur during anaphase, mitotic anaphase or anaphase 2 in meiosis, right. So as over here in this particular cell, what you are seeing is the decondensed form of chromatin material. So prophase yet not begin, right. It started because the centrosome have started moving to poles. That's how we have understood that this particular cell is showing the early prophase, right? Later on, when this particular phase progresses out, what we are going to see is the condensation of chromosome, right? You could see now the X-shaped chromosomes over here, and also you're going to see that it is moving towards it, start radiating the microtubules here, right? The mitotic apparatus formation also occurs during this particular phase, that is prophase, right? So there are various events that occurs during prophase. Those are condensation, disintegration of nuclear membrane and nucleolus, other organelles are also going to be disappeared, mitotic apparatus formation also going to take place. In animal cell, this centrosome movement towards poles also, taking place. So after the prophase is being done and the nuclear membrane completely disintegrate, right? So the disintegration, complete disintegration of nuclear membrane marks the beginning of the next phase, and that is going to be metaphase. So this particular figure over here represents pro-metaphase or early metaphase. Reason being, over here you can see congression. That means the chromosomes that are scattered throughout this particular area are now firstly being attached with the spindle fibers, right? These are the spindle fibers. And also they are now start moving to align themselves towards this equatorial plate. This particular movement is known as congression, right? Now the characteristic event of the metaphase is going to be visible in the next one over here. This is showing complete, metaphase that we see in the books and in the microscope as well, that all the chromosomes are now aligned towards the mitotic equator, the cell equator, right? After this particular alignment is completed, cell progresses towards the next phase, and that is going to be anaphase. So in anaphase, what happens is that the chromatid separation takes place because the centromere is being splitted out. So then these sister chromatids, which now are going to be considered as chromosomes, right? They are going to move towards the opposite poles. And when this is being completed and they reach towards their respective poles, then what happens is that telophase arrives now, and these chromatids, which are now being considered as chromosomes, starts decondensing. Then the nuclear membrane also start reappearing around them, right? Mitotic apparatus is now going to be disintegrated because now it's no more needed. It was there so that the cell could divide properly and distribute, the genetic material equally to each progeny cell. After the telophase ends, this marks the ending of karyokinesis, not the cell division, right? Do remember, never say that it's the telophase that marks the ending of cell division. It is the ending of karyokinesis only. Then after this telophase, the cell moves towards cytokinesis, right? And then of course, on the basis of how the cell is going through the cytokinesis, we can say that we are able to identify that whether it's a plant cell or an animal cell, because both these cells will have different strategy to perform the cytokinesis. Like for example, if we consider, let's say if we consider the plant cell, then in plant cell, firstly, a phragmoplast sort of a structure is going to be created, which is start growing towards the outer side. It's going to create it in the middle, and then it start growing outwards and form the cell plate, which is going to cause the cytokinesis in plant cell. So in plant cell, what's happening, it's from the center, the cytokinesis is proceeding towards the outer sides. On the contrary, if we see the animal cells, over here, the cell starts pinching off like this. So this is called the furrow, So over here, the cytokinesis is being facilitated by furrow formation, right? So sorry. So furrow formation is going to facilitate the cytokinesis in the animal cell. So this is how we could differentiate plant cell from animal cell. One more, criteria for classification. This is another important topic, that how we could classify animal cell from the plant cell. So in the cell division also we see the clear contrasting difference between their, pattern of how they are going to perform the cell division. Like in plant cells, you won't see the formation of aster as they lack centrosome, right? And in the cytokinesis too, what we are seeing that in plant cell, the cytokinesis is occurring via the cell plate formation, and in animal cell, it is occurring via furrow formation. Another important thing over here is that if let's suppose the cytokinesis is not followed by, that means the karyokinesis is not being followed by cytokinesis, somehow cytokinesis fails, then it is going to generate a structure known as syncytium, which basically represents a multinucleate stage, right? So what happens over here, that in a cell, let's suppose karyokinesis is being done, so it has now 2 nuclei, but cytokinesis is not happening. Because of this, these 2 nuclei are going to retain in the cell, and now it's representing the multinucleate stage, and the structure will be called as syncytium, right? So with these, even certain time you will be getting questions which are talking about that what are the characteristic events of these phases. Certain times you will be arranging the sequence not with the names, but rather than the events that occur during a particular phase. So by the event you have to recognize which phase we are talking about, right? So getting back to the question, if we say, so this question is saying that the correct sequence with respect to stages of karyokinesis. So it begins with prophase, where the condensation of chromosomal material take place. So in all of them it starts with prophase, so we can't rule out any of the options. Then prophase is being followed by metaphase, where the chromosomes are going to align themselves with the equator, right? And based on that we could rule out option number 1 and 2, as they are saying that prophase is going to be followed by telophase or anaphase. Then further, metaphase is going to be followed by anaphase, where the centromere splitting is going to take place and the chromatid separates, right? So based on that we could rule out this particular option, as it is saying that metaphase is going to be followed by telophase. Then further, the last phase of karyokinesis, which marks the ending of it, would be telophase, where the nuclear membrane and nucleolus is going to reappear, right? Other organelles are also going to reappear, and the decondensation of chromatin material takes place, right? So the correct option here is going to be option number 4. I hope this is clear to everyone. In case you have any doubts, please put on the comment box so that we can discuss it here. All right. Now moving ahead, we have another question that talks about the significance of mitosis. So when we talk about the significance of mitosis, what all we know about mitosis is, firstly, that it is an equational division, right? So when we say that this is an equational division, it simply implies the fact that whatever number of chromosomes we are going to see in the mother cell will be the same number of chromosomes in the daughter cells as well. Also, how it differs from meiosis is that in meiosis we see crossing over, where the exchange of genetic material takes place. But here no such event takes place, so similar sort of cells are going to be produced, right? So because of this particular advantage that the similar sort of cells are going to be produced, it is basically being used for healing and regeneration. Like for example, if your skin has been broken apart because of an injury, so because of this mitotic division, it would heal back and create the same sort of cells, right? What if it would be meiosis, right? If you would induce meiosis to perform this particular one, there will be certain other type of cells that are going to be produced, and then your entire, viable state is going to be impaired, because now there are different cells which are not being, differentiated for this particular purpose, right. So that is why mitosis, being an equational division and producing similar sort of cells, is helpful for healing and regeneration. So this is one of the significance. Then it says to maintain a proper surface area to volume ratio of a cell. So basically what happens is that when we talk about the interphase, a very big event of the cell cycle, so during that phase, one thing continuously occurs, that is cell growth, right. Now why we are saying that cell growth is continuously occurring? Because when we see different phases of cell cycle like G1 phase, S phase or G2 phase, there is a continuous synthesis of, let's say, RNA, proteins, and in S phase specifically DNA also. So let's suppose if you are talking about this cell and this is the nucleus. Now continuously the synthesis of the molecules are taking place over here, and because of this the cell is getting bigger and bigger, but the nucleus still remains the same, right. A slight enlargement could be there, but the cell growth is enormous, cytoplasmic growth is enormous, right. Now because of this, what happens is that when this surface area is too small, then this nucleus is capable to control all the events, right. The ratio is in a fashion that this nucleus is capable to control the activity of this cell. But when this cell growth occurs and the cytoplasmic growth becomes enormous, then this nuclei is not capable to control all the activities like that, and that is how when the nucleocytoplasmic ratio get disturbed, this is a signal for the cell to divide, right. And the most appropriate method would be mitosis, right. So that is why we could say that it maintains the proper surface area to volume ratio of a cell. So this is another significance. Then the third option says to bring about variation in progenies, right. Variation means whatever sequence we see in the mother cell won't be seeing in the daughter cell. There will be certain difference from the mother cell, but because over here no crossing over takes place, so obviously over here we are not going to see any variations in the progeny cells. But if this question would have asked about the significance of meiosis, this is the significance of meiosis, that it brings about variation in the progenies, right. So this is not a significance of mitosis but meiosis. Now fourth option says that it's a growth of multicellular organism. This is also true, one of the significance of mitosis, because continuous cell division is going to induce the growth in multicellular organism, and the continuous division of creating the similar sort of cells would be done by mitosis. But the question is asking about except one that is not the significance of mitosis, and that's correctly depicted in option 3. Right. So this is how you will be answering this particular question. So after this, let's move ahead to the next question. And this question talks about again cell cycle that most of the organelle duplication occurs in, right. So when we talk about the cell cycle, right, so very small part, less than 5%, would be taken up by M phase. The rest 95% of the time it would be interphase, or we can call it as resting phase 2, because we are not able to see the events that are occurring over here in the microscope, but not because the cell is resting, but rather it works a hell lot, but we are not able to observe it under the microscope, right. So in this particular phase, we could divide it into G1, S and G2 phase, right. So in every phase of the interphase, there is some, kind of duplication or the synthesis is occurring, right. So someone is asking, are these questions PYQ? No, these are not PYQ, but similar sort of questions you could see in PYQ as well, because this particular paper is basically giving you an idea that what kind of questions you are going to encounter in the NEET exam as well. So they are being inspired by that, not the exact ones, right. You may see similarities over here, And this is because you can get an idea that how the questions in NEET appears, right? That's totally fine. I hope you are clear now with the question. Okay. No, they are not entirely PYQs. They are being inspired by it so that you can get an idea that how the questions are going to be appearing in NEET examination as well, right? These are different questions, right? Now, when we talk about, coming back to the question, now when we talk about the different phases of the interphase, in every phase there are certain synthesis is going to occur, and then there will be certain thing that's going to duplicate, right? Like for example, if we talk about the G1 phase, most of the organelle duplication occurs here, right? It is a notable line in the NCERT also. So don't forget, don't skip the lines of NCERT because that's how the questions are being framed. So G1 phase, most of the organelle duplication takes place, right? We can call this particular phase as the post-mitotic phase as well because it occurs after the M phase, right? So post-mitotic phase, over here most of the organelle duplicates. Over here RNA is going to be synthesized, certain proteins are also going to be synthesized. Those proteins which are being required for the very next phase, that's S phase, right? So S phase, also known as synthesis phase, over here DNA duplication occurs, right? This particular event occurs only once in any type of cell division, whether we talk about the mitosis or meiosis, and that occurs only during S phase. So this is a characteristic event of S phase. Now, if we talk about an animal cell where the centriole also present, so centriole duplication also takes place here only, right? In terms of organelle, if we talk about in this particular phase, we are going to see the duplication of centriole. But that occurs only in the animal cell because plant cells, higher plant cells, lack this structure. Whichever cell is going to have it is going to show the centriole duplication in S phase. Now in G2 phase, we see the duplication of mitochondria, chloroplast and Golgi bodies, right? These are the organelles that duplicate during this phase. So this is being considered as the pre-mitotic phase because it occurs prior to the M phase. After this, the cell will progress towards the M phase, which is the actual phase where the cell division takes place, right? So this is how you should know the key notable events of the cell cycle, right? That where exactly the duplication of what is happening, right? So as the question previously asks about, that most of the organelle duplication occurs in, so this is a characteristic line or a notable line from NCERT, that most of the organelle duplication occurs in post-mitotic phase or the G1 phase, correctly depicted in option 4. Option 1 S phase could be ruled out because in this particular phase what happens is, here chromosome duplication, DNA duplication, and in case of animal cell or the cells that possess centriole, centriole duplication also takes place. M phase could also be ruled out. Over here the actual cell division takes place rather than duplication, we could say this division takes place. And over here in G2 phase, we could see the duplication of mitochondria, chloroplast and Golgi bodies, right? So most of the organelle duplication occurs in the G1 phase. I hope this is clear to everyone. Okay, so someone is asking that why cell go in the G0 phase. So if the cell has to differentiate, then it has to break this particular cell cycle in order to get differentiated, because now the cell has to perform a certain function. So in that particular case or in some other stimuli condition, the cell exit the G1 phase, enters into a G0 phase, right? Now this G0 phase, also known as quiescent phase, over here the cell performs the metabolic functions but it won't proliferate until and unless it's not getting back the signals that it has to divide further, right? In case if it gets back the signal to divide further, then it has to enter into the cell cycle. So it gets back again to the G1 phase and start performing the cell cycle. If not, then the cell will remain in the G0 phase, perform the metabolic activities and won't divide, right? So this is how we will be having the description of the G0 phase. I hope this is clear to everyone. All right. Now let's move ahead to the next question, which talks about the respiratory electron transport system, right? We have to choose the incorrect statement regarding that. Now whenever we have to talk about the electron transport system, this is being there in the aerobic respiration so that the ATP could be generated, right? So whenever we have to generate the ATP, the mechanism that explains it is chemiosmotic hypothesis, right? And when we talk about the chemiosmotic hypothesis, we need 4 things, right? 4 requisites of chemiosmotic hypothesis are, firstly, the membrane. Then we will be needing a proton pump. Then we will be needing a proton gradient. And finally, ATP synthase in order to synthesize the ATP, right? As this particular hypothesis talks about that how ATP is going to be formed. So this particular chemiosmotic hypothesis holds true both for chloroplast and mitochondria. But as the question is explicitly asking about the respiratory electron transport system, right, which is basically implementing or facilitating the production of ATP. So we are going to talk about the chemiosmotic hypothesis or chemiosmotic model that operates in mitochondria, right? So this is the electron transport system of the mitochondria. So as I've told you that chemiosmotic hypothesis requires a membrane. So this is the membrane it talks about, right? The inner mitochondrial membrane serves that function of membrane, right? Then what we need are proton pumps that are going to influx it. Yes, so this is what I'm explaining, chemiosmotic theory briefly. So firstly, we are just checking out that whether the requisites of the chemiosmotic hypothesis are being fulfilled or not, right? So the first requisite is the presence of a membrane. Over here in mitochondria, this would be inner mitochondrial membrane, right? The next would be the proton pumps that are going to influx the protons from one region to the other, like they transfer the protons from one region to the other. So over here, there is an intermembrane space. This intermembrane space is basically the space between the inner mitochondrial membrane and the outer one. And this is matrix, the fluid-filled part, right, of the mitochondria. So over here, when we talk about this particular complex, complex 1, NADH dehydrogenase, so it will take up the electrons from NADH, right? So this particular reduced coenzyme gets oxidized over here in the form of NAD+. Why this oxidation is taking place over here is because of the fact that these oxidized coenzymes are being utilized in the Krebs cycle, right? So that the Krebs cycle also, remain functional. So from here, the electrons are taken up by this particular complex, and simultaneously, in order to have this transport done, it influxes the protons inside as well in the intermembrane space, right? So this is acting as a proton pump, that it is taking up the electrons and then influxing the protons over here. Now as we could clearly see, let's say we are making the proton levels over here in both the regions. So as the protons are being taken up from the matrix to intermembrane space, so over here the concentration of protons will be increased, right? And over here it's going to decrease from the original level. Then further, this particular complex gives its electron to ubiquinone. This is ubiquinone. Then this gets reduced and forms this structure, which is ubiquinol, right? This ubiquinone not only takes the electron from complex 1, but also from complex 2, which is this succinate dehydrogenase, right? You have seen this particular enzyme participating in Krebs cycle 2, right? So this particular enzyme is the only Krebs cycle enzyme that is being embedded in the inner mitochondrial membrane, right? Participating in both of them, in the electron transport system as well and in the Krebs cycle 2. So when this particular complex delivers its electron to ubiquinone, it doesn't influx any protons towards the intermembrane space. So this is not acting as a proton pump over here, right? So when ubiquinone is taking up the electrons from both the complexes, it's going to give back the electrons to complex 3, that is cytochrome BC1 complex, right? It receives the electron from both the complexes. These complexes doesn't have any interlinkage. Both of them gives their electrons to ubiquinone, and then finally it reaches to complex 3. Now when complex 3 receives the electron, right, from ubiquinone taking up from both the complexes, it again influxes the proton towards the inner side. This increases further the concentration of protons in the intermembrane space, right? Then this particular complex, when like ubiquinol gives the electrons to this particular complex, it again gets oxidized back into ubiquinone so that it could take more electrons from the two complexes, right? Now this complex 3 is going to give its electron to cytochrome C, right, which is again a mobile electron carrier, and this is being present on the outer side of the inner mitochondrial membrane. Now this takes up the electron from complex 3 and transport it to complex 4, which is cytochrome C oxidase, right? This complex 2, when takes up the electron from cytochrome C, influxes the proton in the intermembrane space, again increasing the concentration of proton over here, right? So we could clearly see that in the matrix the proton concentration is decreasing, while in the intermembrane space it accumulates. That means the concentration is increasing, and that is why, right, that is why we could clearly see that a gradient has been made, right? Over here in the intermembrane space there is a higher concentration, and on the matrix side it will be a lesser concentration. So now we have done with the membrane part. This has been there, proton pumps are there, proton gradients are there, right? These 3 requirements are being done. Now the last one that is needed for chemiosmotic hypothesis is ATP synthase, right? So this ATP synthase doesn't help in the creation of proton gradient, but rather in the breakdown of it, and then the breakdown of this particular gradient is going to create the ATPs, because this particular molecule ATP synthase takes up the protons from the intermembrane space and influx it back to the matrix, and that's how the ATP synthase F1 unit is going to rotate and create the ATPs by combining ADP from PI, right? So this is how the chemiosmotic hypothesis works in case of mitochondria, right? So here ubiquinone is a proton carrier. You could clearly see it's not basically giving up the proton as such. We could consider it as a proton and electron carrier both, because it is taking up both of them, right? So this is how the chemiosmotic hypothesis works in case of mitochondria. I hope this is clear to everyone. Any questions regarding that you could ask? All right, now based on this particular one, we are getting back to our question, which says that we have to choose the incorrect statement with respect to respiratory electron transport system. So the first statement over here says that it occurs on mitochondrial membrane, right? So this is true. It occurs on mitochondrial membrane. The inner mitochondrial membrane is the structure on which this particular respiratory electron transport system works out. Then the second statement says that NADH2 produced in the link reaction are oxidized by complex 1. So this is also true when we talk about NADH, right? We could clearly see that complex 1 is NADH dehydrogenase, right? And what it does is takes up the electrons from this NADH and making it oxidized to NAD+, right? So this particular statement is saying that NADH2 produced in the link reaction are oxidized by complex 1. So it's by link reaction, it could be by Krebs cycle 2, it's going to be oxidized by complex 1. Then third says ubiquinone is oxidized with the transfer of electrons to complex 3. So ubiquinone is already the oxidized form, right? When it takes up the electrons and protons, it's going to be reduced to ubiquinol, right? So ubiquinone is not oxidized when it gives the transfer of electrons, rather it is ubiquinol firstly, which transfers the electron to the complex 3 and it gets oxidized back to ubiquinone. So this particular statement is incorrect. And the fourth statement says that cytochrome is not the complex component of complex 2. So if we see over here complex 2, so in this particular case we could clearly see that there is no cytochrome present. So that is why we can say that cytochrome is not the component of complex 2. This is also right. And as the question demands about the incorrect statement, that's correctly depicted in option 3. So this is how you will be solving the questions. I hope this session is understood by you, and we will continue in the next session. But that's all for this session, and in case of any queries, do post it on the comment section. Thank you very much.